I welcome you all to the Longevity India webinar series. At Longevity India, our goal is to make advanced scientific research accessible to professionals and enthusiasts alike. With this in mind, we are launching our research webinar series to offer deep dives into aging research and technology advancements. Our first speaker in this series is Dr. Ramray Bhat. He is an associate professor in the Department of Developmental Biology and Genetics at the Indian Institute of Science. And he's also an associate professor at the Department of Bioengineering. So today, Ramray will be delving into a crucial topic, which is why aging accelerates the spread of cancer. Ramray heads the Morphogenesis Lab, and his research mainly focuses on the intersection of cancer and morphogenesis, and he uses a lot of computational and experimental techniques to address the various research problems that his lab focuses on. So now I'd like to hand over the floor to Ramri to deliver his talk.
It's been some time since we've done online talks, and but it's fun to get back, and because the power of reach is much higher. I can see some people, some friends here who are not, who are not even in the same time zone. So thank you so much for organizing the talk, for initiating this seminar, Deepak, Suramya, and team. It's a crazy ride, and very excited to be part of the India initiative. So what I'll try to do in the next half an hour or so is to try to give a brief overview of sort of ideas and activities in my lab as part of this Longevity India program. As Amrit mentioned, we primarily are a lab that look at morphological and morphogenetic aspects of what it is to be normal and what it is to be abnormal. In other words, physiological and pathological context. And while we use cancer as a very important context in which to look into these physiological and pathological questions, it perhaps is inevitable and that we have to address the monster in the room, which is aging. And hence the idea to fuse these two together and ask the question, why does aging alter the spread of cancer? And as we all know from our personal experiences, as well as from the epidemiological literature, actually aging happens to accelerate the spread of cancer. So these are preliminary thoughts, ideas, and efforts in the direction of trying to ask this question. All right, so the talk will be sort of broken up into what we know, what we do not know, what we have done, a little bit of what we have done, and a very little bit of what we are doing. And I'll be happy to answer questions on these fronts as we go along and elaborate more. If I keep the at least my presentation somewhat conservative in content. So we'll start from first principles, because that helps me organize my thoughts towards this presentation, which is a first for me on this topic as well. So if we try to understand, strip down of all the molecular details, homeostasis basically is sort of the end point of where development comes to, and development ends with the birthing of a tissue which is organized in the form of cells and its extracellular matrix that surrounds it. And when you have this kind of structure that is achieved through development, by the way, feel free to perhaps ask me questions through the talk, and I would request either Amrit or Deepak to sort of pitch in and interrupt me on people's behalves. So homeostasis, as the arrival point of development, can be understood to be sort of a chronological continuum of the following, where you have basically the microenvironment, the tissue microenvironment, the oops, too many. All right, I'll let others handle the invitations. Homeostasis can be understood to be the continuum of sort of signal, essentially the transduction of microenvironmental cues through signaling within the nucleus through epigenetic regulation of the genome, leading to gene expression, ultimately converging into some kind of a physiological manifestation or physiological expression by the cell, which in turn therefore feeds back into the microenvironment, and hence the environment and the cell keep changing each other, keep responding to each other's cues, and move towards certain function. And it's this function which the cell performs, which is what is regarded to as physiology. Now, we understand large parts of this process through advances in cell biology over the last almost couple of centuries, I would say, of work, and terrific work has been done in trying to therefore develop a systems understanding of this. So if you were to therefore mathematize these terms and bring them together into a form where they can be quantitatively understood, you get what is called systems physiology, or systems biology. And this sort of state, when you depart from the state, this could be called the transformation of this particular tissue state or cellular state, and therefore you get what is tumorigenesis, where you can see how the cells have now altered their shape, size, their numbers, and they are breaking through this environment and trying to move out. So a departure from development leads to homeostasis, or the development ends with homeostasis, and from homeostasis transformation begins. Even in this case, there is with the kind of cancer biology revolution that took place, first in cell biology in the 20s, 30s, 40s, and subsequently with the molecular biology revolution, the discovery of the genes that are altered, we now know considerably also about the systems state of cancer. So we know what are the alterations in microenvironment, how the signaling is rendered aberrant in cancer. There's been a ton of work in the dysregulation of epigenetics. The alteration expression, of course, comes through the reams and reams of transcriptomic and proteomic studies that have been done to show the dysregulation in the proteomes, the transcriptomes of cancer. And ultimately, the pathology is something that has been studied for a long time. And how that pathology feeds back again on the microenvironment is understood through mechanobiological studies, where we now know how the cancer alters the microenvironment, and how the microenvironment alters the cancer. So this also is understood. Now, the interesting part is, from homeostasis, you can have a second route of departure, and that route can be thought to be senescence. Senescence, in the case of the cellular context, is typically looked at as a state where cells stop dividing, which is called replicative senescence. And at this stage, cells sort of move into an altered biochemical state where they tend to start secreting a bunch of interesting secretory molecules. And this state, to a large extent, has also been studied from perhaps the 60s onwards. And while we know, from seminal studies coming from several groups, including in fact Deepak's group, the kind of aberrant signaling that is known to occur in senescence, we know quite a bit about the epigenetic dysregulation that occurs in senescence, and we definitely know a lot about how a lot of molecules are altered in terms of their expression in senescent cells versus their non-senescent counterparts. The pathology still is a little bit of a black box. We, especially in terms of looking at the diversity by which senescence is brought about and the diversity by which it manifests in different tissues, senescence in terms of its pathological tissue state is still a little bit of a black box. Unsurprisingly, even the microenvironmental cues that lead to senescence, or the microenvironmental alterations that senescent cells perform, once they are rendered senescent, is something that is not as well appreciated as the intracellular alterations that are known to occur. Now, coming to the link between senescence and aging, this being a longevity initiative, is regarded as causal at its strongest and coincidental at its weakest. And the reason for this are quite a few. I will talk about a couple of them probably coming in a bit later. But be that as it may, there is quite a bit that is at least understood that with age, we tend to accumulate senescent cells in our systems. And senescence is brought about through several macro environmental cues, cues which could be called, for lack of a better term, as stressors, so stress providing agents, which could range right from what can be called genotoxic stress, such as right hitting at the heart of the cell at its in its DNA, using agents that basically intercalate or cause breaks in the DNA, right up to alterations in perhaps any of these other sort of intermediate points, signaling, epigenetics, which also lead in turn to the cell basically switching off its replicative machinery and starting to alter itself morphologically to tide with such stresses. So therefore, oddly enough, as we can say, both senescence and cancer seem to be a departure from homeostasis. So is aging in some sense. So then it is now being understood, as I mentioned at the beginning, that cancer therefore can well be understood to be again causally or coincidentally strongly associated with aging. And this comes through the epidemiological data that I referred to, where it is seen that for most cancers, the incidence, the progression, the mortality associated with with the cancer, the inability to cope with the treatments, all get worsened as we age. And the age categories at which cancer affects typically tend to be in the later years, as you can see from this graph that came out through a very influential editorial in Nature Aging. So then is therefore, what is the link? And as the title of my talk read, how does aging affect cancer? How does it worsen cancer in so many ways, moving beyond the epidemiological idea? Now, the simplest, the Occam's razor, or the simplest explanation for this has been the what is called the passage of time explanation. As a cell keeps going on and on, or as a tissue keeps going on and on, it tends to accumulate mutations. These mutations lead to, at some point of time, render enough hits to give rise to an a loss in function of a tumor suppressor, a gain in function of an oncogene, and therefore leading to transformation of tissues. And that's the reason why, as we age, we accumulate mutations, and these mutations therefore are sufficient at a certain threshold to give rise to tumor transformation, and that's why we get cancer as we grow old. Now, there is a lot of interesting work that is now being done at using sort of, again, systems biology working across evolutionary scales, which looks at this and has put these ideas into question. And this is therefore the insufficiency of the passage of time explanation, because one has seen that there's this Peto's paradox, which talks about the fact that larger animals therefore should get more cancer because larger animals are fraught through a greater number of cell divisions, leading to a bigger body size. And therefore, if there have been more cell divisions and accumulation of more number of cells, which are now more amenable to more mutations, they should get more cancer. And we see that that's not the case. And further, while Peto did not have the privilege of molecular biological investigations at his end, subsequent work by, more modern work by workers have shown that that Peto's paradox in fact holds. You don't see any strong correlation with body size and tumorigenesis or tumorigenic potential. On the other hand, such studies have thrown up the fact that food habits and other macro environmental factors may perhaps be more strongly correlated. In fact, carnivores tend to have a greater incidence of cancer than herbivores. So hold on to this thought for a moment and keep this in mind as we move along, that carnivores or meat eaters tend to get, or species that indulge in meat eating tend to get more cancer than do non-meat eating species. Now, as we move along, so therefore, this idea of mutation accumulation, this idea of the passage of time explanations for why we get cancer, looks at carcinogenesis itself as a very basic process, as a process that is almost privileges only the cancer cell. And it is well understood that if it was only, it only requires a cancer cell to become cancerous or form a tumor, then you would have even had amoebae getting cancer. It is unsurprising that tumors occur, of course, in multicellular animals. And therefore, one has to understand the process of cancer as not just the occurrence of mutations and the transformation of the cell that gets cancerous, but also to become cancer, to become the disease, to become a spreading disease. It has to overcome various barriers around itself. And therefore, one has to understand cancer as, again, a tissue disease, a disease that can confine the cancer cell if it requires to and keep it silent, keep it dormant, and hence the concept of tumor dormancy. Or, if required, allow the cancer to progress and become metastatic. And this is not new. This has been expressed in various articulate ways by surgeons even from a couple of centuries ago, such as Stephen Paget, who talks about the seed and soil theory, and in a very eloquent fashion talks of the cancer cell as a seed. This is, of course, in the context of colonization, where Paget says that for the seed to grow, you need the right kind of soil. In other words, the secondary metastatic organ has to be the right sort and habitable for the seed to grow in. But one can even talk about this in the context of the organism and say that for a tumorigenic cell, the first cell to become even a growing tumor, a malignant tumor, the microenvironment around it needs to also be habitable enough to allow that. And so if now we look at this sort of microenvironmental understanding of cancer, one can then ask that same question that we asked in the beginning: how does aging or senescence affect cancer, not just by affecting the cancer cell, in other words, the insufficiency of the accumulation of mutation hypothesis, but could aging be altering the microenvironment, weakening the microenvironment, now allowing for the same cancer to grow out in a more florid fashion? And so, computational studies in other contexts by our lab show, using multi-scale modeling, that yes, by altering the microenvironmental cues around tumors, you can not just decide whether the tumor will grow or not grow, but also give rise to different kinetics, different spatial patterns of the way tumors grow and the way tumors manifest themselves, either as collective masses of cells growing or as single cells growing out, and so on. So in that context, I will just talk about some findings of a paper that we did a couple of years ago, well, or a year back, which was published in December of last year in the context of ovarian cancer. So the players in this theater are basically cancer cells, ovarian cancer cells, and I'll put on my laser pointer now. Ovarian cancer cells, which can be assumed to sort of come out from the surface of the ovary and travel to this fluid space called the peritoneum and subsequently settle down. And when they settle down on this sort of layer called the peritoneal boundary, for lack of a better term. And the peritoneal boundary, as you can see, I have drawn. This is, of course, a pre-biorender era, so it's not as pretty as it would have looked like. So these cells are are what are called mesothelia. So the boundary is covered by these mesothelial cells, which cover the boundary and which have to be basically removed, cleared out for the cancer cells to settle down and colonize. So ovarian cancer at its simplest. Now, what we did was the following. I will talk about very few experiments for this talk. I'll try to give an intuitive sense of how we were designing the experiment more than the sort of gory details of the same. What we did was we basically took mouse peritoneal explants or human peritoneal cells and put them down on a surface. Now they are labeled here in red. Now, in one case, they were, I would use these terms a little loosely, so forgive me for it. In one case, they were young, or in other words, control. In other case, they were old or senesced. The senescence was induced using irradiation in one case, as well as in other cases, we used doxorubicin, a DNA intercalating chemotherapeutic that is used in ovarian cancer, incidentally. What we saw was that cancer cells, which were now green and were now sort of sprinkled on this surface, both in the case of mouse as well as in case of the human mesothelial cells. In case of when the cells were not senesced, when they were controlled or again loosely young, the cells were not able to settle down and grow as well as when the mesothelial cells, these are non-cancerous, the red non-cancerous peritoneal boundary cells, when they were senesced, the number of cells, green cells, as you can see from the time lapse as well as playing on the right hand side, the number of cancer cells that attached and grew were far more. So this sort of was the first clue that indicated to us that a senescing microenvironment perhaps is more habitable to the colonization by cancer cells. We then tried to look at what kind of principles might be relevant for this habitability. And that required us to do very close tracking of these cells. So I'm not again going into the details of how we tracked them, but Bharat Thapa, an immensely talented undergraduate and then master's student in my laboratory, an alumnus from the undergraduate ISC program, did some very canny time tracking of these cells and was able to show that using these computer simulations that you need high adhesion of the cancer cells close to the mesothelia, to the senescent mesothelia, disregarding non-senescent mesothelial cells, close to specifically the mesothelial cells for the cancers to grow out. And this can be shown through the simulations where we have non-senescent mesothelia as the sort of slightly pink small cells grow. The senescent mesothelia are the large red cells and the cancer cells are green. So we could see that cancer cells were able to ultimately eliminate the senescent mesothelial cells only when their adhesion was very strong. So this led us to go back to the experiments and ask whether this is indeed true and whether what was mediating this adhesion. So the first experiment that we had done was the senescent associative secretory phenotype experiment, where we took the secretions of senescent cells, and this was done by Mallar Banerjee, in my graduate student in my laboratory. And we tried to look at whether the cancer cells were able to adhere better in the presence of the senescent mesothelial secretions as opposed to the non-senescent mesothelial secretions, and we did not find much of a change. On the other hand, when we took these control and senescent mesothelia, we let them grow and we decellularized them. In other words, we let them secrete their extracellular matrices and then we removed them. And then we sprinkled the cancer cells on top. We found that the cancer cells attached far better onto the senescent extracellular matrix rather than the non-senescent control mesothelial extracellular matrix. Not just that, the cancer cells, once settled down, were able to migrate in a far more directed fashion across this matrix than the non-senescent, than on non-senescent matrix substrata, showing therefore that on senescent ECM, on senescent mesothelial ECM, ovarian cancer cells adhere better and spread faster and migrate better, allowing us therefore a cell biological explanation on why senescence in an ovarian context can allow better colonization of the ovarian cancer cells within the peritoneal cavity, answering in a slightly more cell biological way the larger question of asking how does aging confound, accelerate the kinetics of cancer. Now, moving to just a couple of other sort of slides before I end, one question therefore is that we are often confronted with this idea of senescence being defined through a decrease in proliferation of cells. And it is germane to ask, is decrease in proliferation merely the consequence of senescence rather than even the definition of senescence? Now, this is heretical on some levels, but it needs to be asked because it has been asked by none other than Hayflick. And this particular book on the evolutionary biology of aging by Michael Rose is an immensely interesting read because it goes into those experiments of Hayflick, where Hayflick talks about the fact that even if you take cells from a very old individual and grow them, you still see that they are able to grow several times, several fold more, and then ultimately reach the Hayflick's limit. In other words, cells from senescent individuals still have proliferative potential. And in fact, cells which have been taken out from individuals are even being grown in our labs, showing that they're far outstripped their capacity of aging with respect to the individuals from which they were derived from. This leads us therefore to ask whether decrease in proliferation is just one of the associative properties of senescence, along with other properties of senescence, which must be looked at almost as fundamentally as a decrease in proliferation. And that leads to the question of whether senescence therefore senescence therefore could be construed as a metabolic post-homeostatic state, which includes, among others, a decrease in proliferation. Another important point from this coming from the fact that even post-mitotic cells, senesce, now they are already stopped in their proliferation. So what more can one see in terms of decrease in proliferation? Now, if you recall that point that I made also from the Nature paper, which talked about meat eating being a habit, and this idea of metabolism as an important part of aging and senescence, we then I would like to draw your attention towards another interesting sort of feature that often occurs as we age and as we grow older. We tend to start seeing dysregulations in metabolism that occur within our systems, within our cells. And one common such occurrence is the advanced glycation end products and the dicarbonyls, the volatile dicarbonyls that are basically byproducts, unintended byproducts of the otherwise idealized textbook metabolic pathways, such as in this case glycolysis. And these dicarbonyls, which abound, in fact, are richly present in cooked meat, richly present in diabetic plasma, are involved in cross-linking in a non-enzymatic fashion, cross-linking proteins, leading to glycations of these proteins, glycated hemoglobin being one prime example. And so what we are doing in our laboratories, so this is my second last slide, what we are doing in our laboratories are that we are now trying to look at this competition between the non-cancerous stromal cells and the cancerous cells, and looking at how aging, how metabolic stresses such as that brought about by such dicarbonyls are sort of skewing the competition away from normal cells and towards better colonization by the cancer cells. And the way we do that is by basically labeling these cancer cells, normal cells differently, as I showed you in the previous paper, subjecting them to such kind of stresses, and then watching them basically carry out their competitions. And this last slide basically shows you how we have grown stromal cells in red, cancer cells, spheroids in green. And as you can see, as we increase this dicarbonyl stress, we see that the ability of cancer cells to grow and clear their surrounding stroma keeps increasing further and further. Now, this work has been led by Satyarthi Mishra and Vidhi in my lab, and we now have a series of very interesting factors, which includes loss of viability and decreased cell proliferation, but also a lot of other equally interesting and fundamental cell biological changes that occur in the stroma that allows the cancer cells to grow and defeat them and win the competition evermore. So I'll end with the take-home message that a senescent stroma secretes an extracellular matrix that increases the adhesion and migration of incoming cancer cells. Not just that, aging and chronic inflammation associated with aging creates macro-environmental cues that allow the stroma now to become weaker, allowing for cancer cells to now grow at their expense, clearing them out with brilliant efficiency. With this, I would again thank all the students who contributed to these studies and are contributing the studies as we go along. Immense help to Deepak for intellectual engagement on these topics over the past several years. Prashanjit Mohit, Annapurna from Sri Shankara Hospital, Tillman Glim, my collaborator and long-standing friend from Western Washington University, for working with us on the simulations, and of course funding agencies such as the EMBO Global Initiative Net Investigator Network, the DBT Wellcome Trust, India Alliance, the John Templeton Foundation, and of course the Longevity Initiative, which is funding some of the work we are doing towards trying to understand this very interesting dialectic between cancer cells and aged non-cancerous defending tissues. Thank you so much.
Yeah, thank you so much, Ramray. That was really a quite illuminating talk, I would say, and a great start to our first webinar series. So I open the floor for people to ask questions. Your microphones have also been enabled in case you want to ask Ramray a question directly, or else you can also drop in any questions you have in the Q&A section of the chat box. Yeah, so in the meantime, I do have one question to ask you, Ramray. So your observations about the enhanced, migratory capacity of these cancer cells in the senescent mesothelia. So that also kind of, uh, makes someone curious to think whether, recurrent tumors, because let's say cancer cells themselves can become senescent. So once
Yes.
you once you hit, let's say, a chemotherapy drug that causes DNA damage and then causes senescence, that again could explain, let's say, an increased aggression in recurrent tumors and so on.
Right. Yeah, Amrut, that's a great question, and I think again one needs to sort of take the bull by its horns by looking at the role of senescence in once again tweaking the inter niche cooperativity within tumors, because within tumors we know, for instance, in the the second story that I talked about, which I just referred to its beginnings, but we have the end in sight. We know, for instance, if the dicarbonyl stressors, the cancer cells seem to be very efficient at bypassing this stressor by having the right kind of enzymatic machinery to basically dissolve their potency. And so cancer cells will therefore again have variant abilities to become senescent. And so within a tumor, you may have senescent tumor cells and non-senescent tumor cells. Now, how that alters their intercooperativity, whether you need a certain senescent tumor population to also increase the migration of non-senescent tumor population, is something that needs to be looked at and studied very carefully in the coming days. But it also sort of talks about how tumor cells may be senescent at the same time proliferating. Since I tried to sort of dissociate the proliferation phenotype from the senescence phenotype, and leave you with a sort of metabolic, perhaps narrative of senescence, there could be, therefore, now one can think of multiple different senescent cells also coexisting within a tumor, having their own different properties, in turn contributing to tumor heterogeneity. So there's a new axis of tumor heterogeneity, and that of senescence could be looked at in the future.
Yeah, thanks. Great. Looks like we have a couple of questions. So, Shahid, you can go ahead.
Oh, hi, Ramray. Can you hear me?
Yes, I can hear you, Shahid. Adab.
Oh, it was really nice hearing your talk after so long. So it was very fantastic talk, and I really liked it. I have a couple
Yep.
of more general questions regarding your where you showed that Met5As where you senesce them, you made them senescent cells by irradiation. I was thinking that did you try any other ways of making them more senescent, like senescent, and then see the same phenotype?
Yes, Shahid. So the cells were made senescent in two ways in this publication. We used both irradiation as well as doxorubicin. Doxorubicin as a sort of a genotoxic agent. Incidentally, it's a very interesting sort of discussion of the paper that we had, where we naughtily put out that therefore it looks like even chemotherapeutics may not necessarily be doing all good things in their abilities within mixed tumor microenvironment, and they might, an excessive and overuse of chemotherapeutics may hurt us in severe ways by actually allowing dormant tumor niches to now sort of come out. So yes, we used doxorubicin, and in the coming days, with funding from the Longevity Initiative, we are testing out more different stressors which lead to senescence, and we are looking at how these different other sort of more, I would say, softer stressors and or inducers of senescence also allow us to look at how these things are playing out.
Thank you. Um, so the next question which I wanted to ask you, so you ultimately saw there is a difference in secretome, for example, the ECM that these senescent
Yes,
cells secrete.
yes.
Do you had any chance to look at the ECM
Yes.
or
So
what these senescent cells?
yes,
Yeah.
yes. So we looked at it in a more targeted fashion, and we see hyaluronic acid come up in the senescent matrix. We see that fibronectin comes up in the senescent matrix as well, to a larger extent than the control mesothelial matrixome. So we know that a couple of candidates, in fact, to that extent, we even used an RGD cyclopentapeptide to prevent the adhesion of the senescent, the cancer cells. And we see that when we add the RGD peptide, the adhesion of cancer cells to non-senescent matrix and senescent matrix almost becomes the same. So that told us
Oh,
that fibronectin is playing an important role in this whole process. I'm so sorry. Yeah.
I see.
So yes, we know that fibronectin and hyaluronic acid are definitely upregulated, but more sort of unbiased proteomic studies need to be done to identify
I
other novel players.
Yeah. Thank you so much. Thank you
Hi,
so much.
Thank you.
can I ask?
Yes.
Hi, my name is Dr. Janardhan. I'm a gastrointestinal surgeon, particularly interested in colorectal cancer. My sort of question to you is, do you find a linkage between the incidence of cancers such as ovarian, breast, colorectal, and those sort of cancers than others? Because what I'm coming to is, say, for an example, you take a colorectal cancer, it seems to be much more common, as you know, in the rectum and in the
Yes.
sigmoid area.
Yes.
So do the cells start as a single cell, or there's an environment in which a cell changes?
Right. In case of, so this is, you lead me to some very interesting territories. So in case of typically for colorectal cancers, since there's been a lot of very good genetic studies, but especially associated with the West, where it afflicts, further up in the colon. It's generally thought to at least originate, as a single cell rather than a collective. However, the colonization or the spread occurs through these cancer collectives, which then sort of, break through the serosa and then enter into the peritoneal cavity. As far as the rectal, manifestation is concerned, which is something which we see more in India, all bets are off. I don't think the same kind of genetics prevails for the rectal variety. And there is much work to be done in that direction towards trying to decipher the cell biological behaviors. Incidentally, since you mentioned colorectal cancer, there's been, again, although it may start from a single cell, we know that there is something called tumor budding that was looked at by German pathologists right in the 60s and 70s, where you could see that even within the stroma, or in the submucosa, you would see collectives of cancer cells sort of moving in almost synchronously, harmoniously in one go, and that was called tumor budding. And there's a fair amount of work that's coming out of Institute Curie on how this sort of behavior comes through. Again, whether that prevails in the case of rectal cancers or the rectal variety of colorectal cancers is something that could be very rigorously looked at histopathologically. Not been done.
Yeah. To take it a step further,
Yes.
Do you have any answers as to why in certain colorectal cancers you find the metastasis has happened earlier, in others you find a large tumor, but still there are no mets in the liver? Now, I'm a surgeon, so that's why the scientific basis is very little for me, but it's just a practical thing.
I understand. I'll be sticking my neck out in speculating on this, given that neither am I a surgeon nor am I a biologist who looks on colorectal cancers.
But you are the ones who had to give us the answer.
Yes. But given the fact that one needs to therefore look again at the Paget's seed and soil idea to look at what are the genetic predispositions that allow for a tumor to grow as a collective, but still not invade into the peritoneal cavity. Whereas in case of certain others where you do not see a large mass at the primary focus, you might still get rank metastasis. In fact, work has been done, at least in breast cancer, where one has seen that the progression is of cancer through the metastatic trajectory can be fairly non-linear in terms of single cells escaping out of the primary tumor even before the primary tumor has reached discernible volumes. Single cells shedding out and reaching a secondary site and metastasizing it. So in other words, the trajectory can be fairly non-linear. And we can discuss more about this perhaps at a later forum.
Thanks. So we have Shiva who has his hand raised. So please go ahead.
Yes, sir. Sir, I was asking, just as you said that the senescent cell still have potency to proliferate if we put it in different environment. So I want to ask that if the cancerous cell, if it moves to healthy tissue, how does it affect? Like there the tissue is healthy, then like how does it grow there properly?
Yeah. So I'll try to answer that. I did not say that senescent cells can regain proliferative capacities. In fact, what I said was that the association of the loss in proliferation and senescence need not be causal or definitional. That was the only point that I was trying to make. Now, coming to the point you make, this is something that is very interesting, and it relates to this particular point, the senescence aspect, that we see that if you have an epithelial monolayer, a layer of healthy epithelial cells, you don't need immune system and 5 other systems to actually defeat cancer or prevent cancer from taking root. And a strong elastic epithelial monolayer can itself very nicely prevent cancer spheroids from attaching or growing within them. And so the idea of clearance of cells by cancer cells is in some sense to be questioned and problematized and looked into it. It is only when this layer of cells is stressed using either senescence or perhaps some other metabolic stress that the cancer is now able to take root. Otherwise, a simple epithelial layer is able to very nicely keep cancer at bay. In fact, this is the reverse of what some people call epithelial defense against cancer, in which if you have a cancer cell within a mass of a sea of epithelial cells, they can push the cancer cell out. Just the way they can push a cancer cell out, if a cancer cell approaches them, they can prevent the cancer cells from also going in. Okay, but again, like I said, only when you stress this layer, either due to aging or due to metabolic alterations or due to chronic inflammations and so on, which stresses which the cancer cell itself can take care of, under those conditions, the cancer cell is able to take root and grow. Was
Thank
I
you,
able
sir.
to make myself clear? This is,
Yeah,
of
yeah.
course, relating to certain tumors. There are, like I said, there are pediatric cancers and pediatric tumors as well. So the cancer can grow even within younger tissue microenvironments. But those are rare, much lesser in comparison to the age association of cancer with most cancers.
Okay, sir. Thank you.
Thank you.
We have one question in the chat box from Parmar, who is asking for your take on the hypothesis that senescent cells are an anti-cancer mechanism to prevent uh, conversion of cells to a cancerous phenotype.
Right. So yes, this idea has been around for that senescent cells in some sense are like I said, an additional route away from homeostasis. So if one can think therefore of a cell having to decide between a homeostatic and a senescent versus a tumorigenic fate, and if it therefore takes a senescent route, it prevents itself from therefore accumulating enough mutations to become tumorigenic. So to that extent, this particular sort of bifurcation is very much prevalent. At the tissue state, however, how that manifests remains to be seen, given the fact that, as I said, if you have some cells becoming senescent, and if you have some cells becoming transformed, the senescent cells may even aid transformation, the transformed cells. So it depends on the cue and the context in which you look at this particular idea. And I think the idea has to be spatialized. One cannot look at a cell, whether it comes to senescence, whether it comes to cancer, whether it comes to homeostasis. One has to look at the tissue state and examine the tissue state very carefully.
Thank you. We have a question from Ananya Sarkar, who asks, in case of senescence models, are you considering biological or chronological aging in a broad view?
Again, I think if one were to look at it from a metabolic point of view, it wouldn't matter whether we are looking at a chronological or a biological one, but we primarily work with the biological models of aging, where we look at stresses. These stresses can naturally accumulate with time, or they can attack even a younger environment. In fact, if one looks at a microenvironmental mechanism of senescence, then one need not even wonder about this question so much or parse between such models, because the microenvironment can either age or it can assume aspects that mimic an aged microenvironment.
Right. So thanks, Ramray. I think that's about all the questions that we have for today. Okay, wait, there's one more. Okay, yeah, that's it. Thank you. Yeah.
Thank you so much. Thank you.
Yeah. So thank you, everyone. And thank you, Ramray, once again for hosting this amazing talk. And we look forward to bringing you more talks about aging and longevity in the near future. So keep your eyes out for the Longevity India research webinar series. Thank you, everyone.
Thanks all. See you.